Welcome to our second topic in Biology 2301, Human Physiology. This week we're going to be learning about the nervous system and how our information and our knowledge on ionic movement across the cell membrane and the use of protein channels is really going to help us to understand how nervous physiology works. We're going to be learning about the components of the nervous system as well as specifically how the nervous system is able to generate electrical potentials and send those electrical messages around our body. A little bit of background begins with Objective 7. The nervous system is the master controlling and communicating system of the body. In general, your nervous system has three functions. It uses sensory receptors to monitor changes, which we call stimuli, that occur inside and outside of the body. This gathered information is called sensory input. So if you put your finger on a burner and you feel that, that is a sensory input. If you can hear my voice, that is sensory input. Your nervous system also integrates the information, which means it processes it, analyzes it, interprets, makes decisions about a potential response. It can also store the information. Thirdly, your nervous system causes a response called a motor output by activating effector organs, which can be muscles and glands in your body that act in response. When you pull your finger away from that hot burner, that's a motor output. The nervous system sent a message to a muscle which contracted to move your finger away. That's a response sent on motor nerves. This is a nice pathway of how the nervous system operates in a very simplistic form. In this case, you may be thirsty. You may want to pick up that water. You see that the water is there. That's sensory input. And that information of vision or thirst is sent via a sensory neuron toward the brain or CNS, which will integrate the information. And it says, okay, let's use muscles to pick up that water. And so it's going to send information out via motor output or a motor neuron to a muscle that will allow us to complete an action. This is very simplistic, but we do this every moment. We are monitoring the environment at all times. We are analyzing that environment, and we are bringing about decisions or change based on that. Now let's organize the nervous system. The nervous system has two main divisions. That is, the central nervous system, or CNS, the brain and the spinal cord, and the peripheral nervous system, or PNS, which includes all of your nerves, spinal nerves and cranial nerves, as well as your sensory receptors. Your central nervous system, as well as your peripheral nervous system, work in conjunction with each other, and we're going to take a look at a few more divisions. When we talk about the central nervous system independently, this is your integrating center. Your peripheral nervous system would be both the sensory input as well as the motor output. Let's take a look at another divisional layout. Again, we see that the central nervous system includes the brain and the spinal cord, and that again is our integrative and our control center. This is where we analyze the information. Your peripheral nervous system includes all of your nerves, cranial nerves, spinal nerves, as well as those sensory receptors. This provides a communication between the CNS and the rest of the body, so information coming in and information going out. The information coming in is part of what we call the sensory or the afferent division. You learned that afferent means toward the CNS. We have sensory or afferent divisions that bring sensory information in from somatic and visceral sensory nerve fibers. Think about somatic being skeletal or skin, and visceral being internal organs or involuntary structures. This sensory information is going to be conducted toward the CNS from receptors that you have around your body. The decisions that come out of the CNS would be motor or efferent. Efferent means away or traveling away from the CNS. And these are motor nerve fibers. Motor nerve fibers are conducted as impulses from the CNS to effector organs. An effector organ would be an organ that can respond, can do something. And muscles and glands are actually able to do something. They can secrete or they can contract. So based on that, since we have different kinds of muscles and glands, we will have two divisions of that motor output response division. And those two divisions are the autonomic nervous system, or the ANS, and the somatic nervous system. The somatic nervous system are voluntary structures. These are somatic motor structures. Structures that you can voluntarily control with a motor nerve, like your skeletal muscles. So any impulse that comes from the CNS to a skeletal muscle is part of the somatic nervous system. The autonomic nervous system is involuntary. It's also called your visceral motor. These are impulses that are conducted from the CNS to cardiac muscle, smooth muscle, and gland. What do those three structures have in common? Well, you don't control them. So the autonomic nervous system, your involuntary nervous system, is part of a motor output response to structures that are involuntarily controlled. Since you don't have conscious control of these structures, we have two more divisions that will work together to control and to regulate, keep in balance your autonomic nervous system. And that is your sympathetic and your parasympathetic division. Your sympathetic nervous system is an involuntary nervous system that acts as an emergency response unit. I always think of fight or flight. This mobilizes your body systems during an emergency. When you run away, when you're worried, when you're anxious, when you're nervous, when you're afraid, when you're excited, when you're exercising. Your sympathetic division, if you recall, is also part of that division which controls pupillary responses. In this case, it would cause pupillary dilation. The parasympathetic division is in opposition. If the sympathetic turns it on, the parasympathetic would turn it off. This is more of a rest and digest system. This is for non-emergencies. Think of being more relaxed. Lowered heart rate, lowered respiration rates, lowered blood pressure, but we do turn on digestive functions. We also learned back in anatomy that the parasympathetic division also has function in the pupillary control, in that it actually controls pupillary constriction. Now that we have a better idea of how the nervous system is organized, let's take a look at some of the cells that comprise the nervous system. Let's take a look at histology. The nervous system's histology, or the basic tissue of the nervous system. Recall that again, the nervous system allows for communication between cells through sensory input, integration, and motor output. How does it do this? Well, the tissue of the nervous system only uses two overall categories of cell types. Those are neurons, which you see pictured, and neuroglia, which are much more of a support cell. Remember that a neuron is the structural unit of the nervous system. That means that when we say that the nervous system is able to conduct electrical messages from one place in the body to another to monitor the sensory environment and to bring about change, all of that function is brought about by this particular kind of cell, a very unique cell. Neurons are highly specialized cells that can conduct these electrical messages from one part of the body to another. They form long chains integrating and interconnecting every part of your body. These very, very unique cells have extreme longevity, which means they're going to be as old as you. You stopped forming them, however, when you were about 2 years old, and so since then you haven't made any new. You may make new extensions, but no new cells. And so this amitotic problem, if you will, for nerve cells means that we have to protect the few nerve cells that we have. These cells eat a lot, they breathe a lot, if you will, also. They have a very high metabolism. They're very active. And so we have to treat them well, feed them with good food and good oxygen. As a general overview, a neuron structure is pretty typical. What you're looking here are 3 different kinds of neurons that you may see in the body. Here we have a typical motor neuron, which is what we'll study the most. This is a pretty typical interneuron, as well as potentially a sensory neuron, as well as this is more of an interneuron or sensory neuron. You can just see a slightly different shape of the cell body and the type of extensions which form the chains throughout the body. The neuron will have a cell body, and that cell body is where most of the organelles will be housed, as well as the nucleus. This cell body also helps us to bring information in that's both excitatory postsynaptic potential, and inhibitory postsynaptic potential. We're going to talk about those quite a bit more, especially when we talk about the dendrites. The dendrite are extensions off of the cell body that are very, very short, but their job is to bring the information in, both excitatory and inhibitory, to the cell body. What you see here is a long axon which extends off of that cell body. Most axons are single and they don't branch until their very terminal end. Let's take a closer look. As you see here, this cell body, we also call this the biosynthetic center of the cell, and it has all the organelles that a typical cell would have, except no centrioles. Without a centriole, a cell cannot undergo mitosis, no dividing, and that's why I mentioned that you developed all the neurons that you currently have by the time you were 2 years old, and then no more divisions going on. So if we do lose some neurons, we can't gain any new. Off of that cell body, you have these multiple short extensions, and these are called dendrites. Dendrites are the receptive region of the neuron. They're able to receive signals from other neurons or from stimuli. They're able to conduct their electrical signal toward the cell body. The signals are not nerve signals, but are called a graded potential. These short tapering processes are very numerous, heavily branched, and close to the cell body. They also contain all the organelles found in the cell body. As I mentioned, the graded response can be both excitatory or inhibitory. We're going to talk about these types of responses and types of electrical potentials very soon. The axon of a neuron or nerve cell is a single long branch off of that cell body. They are considered to be the conducting component of the neuron. Axons generate nerve impulses, now called an action potential, and transmit them typically away from the cell body, often over very long distances. This region right here, which looks like a funnel, is called the axon hillock, where the axon first extends off of that cell body. The axon hillock is a cone-shaped region that actually is also called the trigger zone, because that's where the action potential is very first generated. Let's move down that axon, and you see it terminally branches. This terminal branch, the branches themselves are called telodendria, but at the telodendria end, we have little feet called axon terminals. Axon terminals are the end bulge of an axon that is able to store and release a chemical called a neurotransmitter, and this will elicit the effect in the next cell of our neuron chain. You may notice that axons are often myelinated. Myelin sheathing is a protective coating, as you see here in blue. Myelin sheathing is a lipid covering of long axons that acts to increase the speed of the nerve impulse conduction. It insulates the neuron and helps to guide regeneration in the PNS if potentially a nerve axon has been severed. The myelin sheathing is a lipoprotein, lipid and protein that acts as an insulator around many of our long axons. How do we form a myelin sheath? Each of these cells is called a Schwann cell. It's one of our neuroglial cells or support cells. These are neuroglial cells that are able to wrap around the axon and create the myelin sheathing in the peripheral nervous system, or PNS. A similar myelin sheathing is capable of insulating a neuron in the CNS, but uses a different type of support cell called an oligodendrocyte. When cells line up and wrap sections of our axon, you see that there are gaps created in between each adjacent cell. Those gaps are called the nodes of Ranvier. Those become very important as we look at saltatory conduction. Saltatory conduction is conduction of a nerve impulse that jumps from node to node. We're going to learn how electrical potentials begin here in the axon hillock, and that electrical message can be sent along the length of the neuron. We're also going to learn that it has a lot to do with ionic movement across the cell membrane. The reason that the myelin sheathing is able to increase the speed of transmission is because the electrical message can simply jump from one node to the next, to the next, to the next, covering less surface area and actually moving faster. Most of our very long axons, specifically in our limbs, will be myelinated to increase the speed at which they can send a message. Shorter axons, for example, in your brain perhaps, don't need to be myelinated because they don't have to travel the long distance and the speed is not as important. Let's take another look at those axon terminals. The terminal end of an axon is branched, and at the very, very end, little bulbs or feet are called axon terminals. They can also be called synaptic knobs or boutons. These are heavily branched termini of the axon. The boutons are the bulbous distal end of the telodendria and are the secretory component of the axon. The neurotransmitters, chemicals, are synthesized, stored, and released from this axon terminal. When an impulse traveling along an axon reaches the axon terminal, it causes the release of neurotransmitter into a gap called the synapse, which triggers the next cell in our chain. Let's take a look at how that will happen. We mentioned that neurons do occur in chains throughout our body. For example, again, putting your finger on a burner elicits a response in a sensory neuron, which would send that information to the brain. The brain would integrate that information with an interneuron, and then send a response via a motor neuron out to our muscles to allow us to move our hand away. In order to send a message from one cell to the next, that electricity that goes through the cell is chemically transferred. We wouldn't want electrical message to jump from one cell to the next, electrical message jump one cell to the next, because we could lose that electricity. So what we do is that electrical message gets sent to the axon terminal. At the axon terminal, we release chemicals, and those chemicals create the electrical response in the next cell. The gap between the two cells is called the synapse, or the synaptic cleft. The neurotransmitter is released into this space. The neuron that comes before the synapse is called the presynaptic neuron or presynaptic cell, And the neuron that comes after the synapse is called a postsynaptic neuron or postsynaptic cell. We will cover a detailed view of the synapse and neurotransmitter transmission, but not until later objectives. As a slight review, I also wanted to put up this PowerPoint and show you that all of the different types of neuroglia, which you did learn in your anatomy class. Remember that astrocytes and microglial cells, ependymal cells, and oligodendrocytes are only found in the CNS, with the oligodendrocyte forming the myelination. Schwann cells and satellite cells are only found in the PNS, with the Schwann cells forming that myelination. I mentioned that you form all of your neurons as a toddler, and from then on, our job is to protect them, increase the connectivity between neurons, and house them until we die, hoping that we don't lose too many as we age. Many times we do run into times of injury, where an axon may be severed or cut. Surgeries, wounds could sever or cut an axon. So this small objective talks about the possibility of regenerating a delicate, specialized neuron in the case of injury. If an axon is severed, they could repair themselves and restore significant function, provided that the damage occurs outside of the central nervous system and does not affect the neuron cell body. These are provisional importances. The peripheral nervous system has a unique way of regeneration that we're going to walk through. Damage to nervous tissue is serious because mature neurons are amitotic. As we've mentioned, they do not divide, so you cannot make any new. If the damage is severe or occurs close to the cell body, the entire neuron may die, and other neurons normally stimulated by its axon on down the chain may die as well. However, in certain cases, a cut or a compressed axon on a peripheral nerve could regenerate. So it's important that our site of injury is not close to this biosynthetic center. If our injury is far enough down, and usually a fairly clean cut, not too much damage, we can begin the process of regeneration. Immediately after the neuron axon is cut, in the PNS only, the two ends of the site seal themselves and swell as substances being transported along the axon begin to accumulate. So you see the swelling here. The distal end degenerates. This is called Wallerian degeneration. Schwann cells, macrophages all phagocytize the disintegrating axon and the myelin, which means they break it down. So the entire axon distal to the injury is degraded, but our neurolemma, which means our Schwann cell encasement, may stay intact. This is wonderful if it can stay intact, as it may guide the regeneration. The portion of the axon still attached to the cell body will give rise to a growth cone, which grows out to an effector organ very, very slowly, potentially only about 1 millimeter per day. Schwann cells that may have remained intact will guide the regenerating axon as kind of a scaffolding that the axon could grow through. The Schwann cells form cellular cords that guide the regenerating axon, form sprouts across the gap to their original contacts. The Schwann cells can also release growth factors. It's called NGF, or nerve growth factor, to encourage this axonal growth. Here is the downside of neural regeneration. Let's take a look at regeneration in the CNS, or central nervous system. Regeneration in the central nervous system never occurs in distances more than 1 millimeter under normal circumstances. Of course, we've heard stories of exceptions. Damage to the brain and spinal cord has been viewed as irreversible, due mainly to the support cells in the CNS called the microglial cells. After the central nervous system is injured, the microglial cells and astrocytes rid the area of any deteriorating cell debris. The oligodendrocytes also die and fail to oversee and guide the fiber regrowth. The dead oligodendrocytes are replaced with a glial scar, which in fact blocks any sprouting axons. Also, myelin sheathing of nearby axons may contain a growth-inhibiting protein. Future research ultimately is trying to reverse the effects of CNS regeneration blockade. But for now, the microglial cells and the astrocytes tend to confound any type of regrowth. Now that we've talked about nervous histology and a basic overview of nervous organization, I'd like to talk to you about how it is that nerve cells are actually able to not only receive an electrical message, but generate electrical messages and conduct those electrical messages or signals throughout your entire body. If you ever saw the movie The Matrix, they kind of related us to being a battery, which is true. We are very energetic. We can create electrical messages. So what we have to do is talk about how that's possible. We're going to set up the situation, talk about polarity one more time, and then talk about how we can generate these very important nerve signals. A couple things that we have to remember from week 1. Recall that the predominant solutes in your extracellular fluid are sodium and chloride, which means in the fluid outside of a nerve cell or any cell, the predominant amount of solute is specifically sodium and chloride ions. Now, the intracellular fluid, the fluid inside our cell, also contains a high concentration of sodium chloride, but also potassium ions and non-diffusible molecules, specifically a negatively charged protein count. Electrical phenomena result from the distribution of these charged particles that occur at the cell's plasma membrane and play a very important role in how signals are integrated, conducted, and how cells communicate. This means that ion movement across the cell membrane has everything to do with how we generate the electricity needed and how we are able to conduct that electricity over long distances. An electrical potential is separated electrical charges of opposite sign that have the potential to do work if they are allowed to come together or in opposition. This potential, called the electrical potential, is measured in volts or millivolts. What that means is what you're looking at is a cell membrane with opposite electrical charge on outside the membrane and inside the membrane. This opposite electrical charge creates a polarity, and that polarity is allowing us to potentially create a spark of electricity, which will be a nerve signal sent throughout the body. Let's take a look more closely at the plasma membrane of a neuron to see how it's able to both generate and conduct electrical signals or action potentials. One thing we have to remember is that even though this cell doesn't look like a typical cell, it has a very typical plasma membrane. And a plasma membrane, recall, is composed of a phospholipid bilayer and the very important embedded channel proteins within. Here's a close-up of our plasma membrane, where we're going to take a quick review of the types of protein channels that we will have that help to control the amount of ion on the outside and the inside of our neuron. Remember that the plasma membrane of a neuron contains ion channels, which are made of these membrane proteins. Some are always open, they're called leakage or passive channels, but others have a molecular gate, which means something must unlock that gate to allow the chemical to go through. Gated channels respond to some kind of signal that can open or close the protein channel. That signal could be a chemical, like a neurotransmitter, which could open the gate. It could also be opened based on a change in voltage. Here's a good example of a voltage-gated channel. You see the positive charges outside the cell membrane and the negative charges inside the cell membrane and a closed gate. Let's reverse those charges and make it negative outside and positive inside. Now you see that gate is open and sodium can now go through. The difference is, here's a chemical gate, closed, but when a chemical binds, like a neurotransmitter, we open the gate. With voltage gated, a change in potential or charge at that membrane can open the gate. Your neuron has both types of gates in the membrane at very unique locations. This is an important one to remember. You will find chemical gated channels, which are also called ligand gated channels, within the dendrites and the cell body of a neuron. Voltage-gated channels are unique and specific only to the axon hillock and the axon. When channels open, what you're looking at is a little rendition of our cell membrane with various types of ions inside and out. Recall that ions move along their chemical gradient when they diffuse passively from an area of higher concentration to an area of lower concentration. Ions can also follow electrical gradients when they move toward an area of opposite electrical charge. When a gate opens, an ion channel opens, ions can diffuse quickly across the membrane following both their chemical gradients and their electrical gradients. This creates both an electrical current and a voltage change across the membrane. Let's take a look a little more closely. In order to generate a nerve impulse, also called the electrical signal or the message being sent, in order to do this, we have to set up our cell in a resting position. Consider that a ready position, and that's called the resting potential, or the RMP. The resting potential is the polarized state of the cell. This is a position, a time when the axon is not conducting a nerve impulse, but again is in the ready state. In this time, there are more positive ions outside the membrane than inside. This can be measured as a negative 70 millivolt charge inside the axon. This is created as more sodium is outside the cell and more potassium is inside the cell. Let's explain. This measurement across the membrane is a relative term, saying that the inside is more negative than the outside. All cells at rest have a potential difference across their plasma membrane, with the inside of the cell negatively charged with respect to the outside. Again, this is called the resting potential. Remember that we have a lot of sodium kept outside, we have a lot of potassium inside, but they're both positive ions. We have way more sodium out than we have potassium in. We also have many negatively charged ions inside the membrane that cannot diffuse, which add to the negativity. This creates a positive environment outside, a negative environment inside, which can be measured at negative 70 millivolts. That's an important number. The resting membrane potential has to be in place in order to generate a graded potential or an action potential, or the current that allows for cell-to-cell communication. We're going to see that in order for one cell to talk to the next, in order for you to wiggle your toes or think a thought, we must first have an electrical message that begins with a graded potential and then stems into an electrical potential. These are steps to generating that message. At rest, the resting membrane potential, the cell cytoplasm contains less concentrations of sodium and higher concentrations of potassium than the extracellular fluid. So you see, there's more sodium outside than in, and there is more potassium inside than out. There is a high concentration of negatively charged ions that are proteins as well. Why does this occur? How do we set this situation up? Two ways. First, this is due to the permeability of the plasma membrane to sodium and potassium, the natural permeabilities that we'll talk about. It's also due to the sodium-potassium ATPase, which transports sodium out and potassium back in using ATP as energy. Permeability first. A membrane at rest has very low permeability to anions, so negatively charged ions inside the cell may not diffuse at all. They're stuck in there. It is slightly permeable to sodium, so very little sodium is actually able to get in, but it's freely permeable to potassium. You see the larger arrow. Therefore, potassium diffuses out following its concentration gradient faster than sodium can diffuse in. The net result is there is more positive ion leaving this cell than coming in. This leaves the cell interior with a more negative charge. Potassium can use leak or passive potassium channels that are always open. Sodium can leak as well, just so much more slowly. That's one of the ways that we set up this positive outside, negative inside condition. The next way we set it up is using the sodium-potassium pump. The ATP-driven sodium-potassium pump will eject 3 sodium to every 2 potassium coming in. The ATPase pump pumps in or out the exact amount that leaked through those open channels. This stabilizes the resting membrane potential by maintaining the diffusion gradients for sodium and potassium. Since we are pumping more sodium out where it's already very positive, and potassium in where there's already very much potassium, of course we're going against the concentration gradients, and so ATP is required. This is helping to establish the very positive exterior and the very negative interior, as well as establishing the chemical gradients that sodium and potassium would want to follow. This is how the neuron appears when it is inactive. If we want to send an electrical message, we must reverse the resting membrane potential. I think this is a great place to stop. We'll stop this first video now, and then we will pick up with the idea of how to reverse that resting membrane potential and create the electrical charge that's going to send a message down to our toes and back.